My dear students, Assalamu alaikum to all of you. Physiology of muscle contraction for class 11th. This is the physiology of muscle contraction or mechanism of muscle contraction. According to the principles of physiology of muscle contraction, the laterally projecting heads cross beads of the thick myosin myofilaments come in contact with the thin myofilaments and rotate swivel on them. This pulls the thin myofilaments toward the middle of the sarcomere past the thick myofilaments. The Z lines come closer together and the sarcomere becomes shorter. Length of A band remains constant. A similar action is in all the sarcomeres results in shortening of the entire myofibril and therefore of the whole fibril and the whole muscle. A contracted muscle becomes shorter and thicker, but its volume remains the same. There is a theory for the muscle contraction that is called as sliding filament theory. According to this sliding filament theory or sliding filament theory for muscle contraction, this theory has been put forth for the understanding of the mechanism of muscle contraction. The above view about the contraction of straight muscle is called the sliding filament theory. It states that muscle contraction results from sliding of thin myofilaments past the thick myofilaments. There are thin myofilaments and thick myofilaments. The sliding filament theory suggests that these thin myofilaments slide over the past thick myofilaments. What are the myofilaments? These myofilaments are the actin and myosin protein fibrils. They can be shown as here. These myofibrils are just like these, what we call as this is tropomyosin, and this is actin, and these are calcium double positive ions, and this is troponin protein. So far the mechanism is concerned, sliding filament theory. The muscle contraction results from sliding of thin myofilaments past the thick myofilaments rather than by shrinking of myofilaments. These myofilaments do not shrink. They slide past one on each other. That is why the muscle contraction takes place. This theory was proposed independently by A.F. Huxley. A.F. Huxley was the first scientist who put forth this theory. And one more scientist is there, Niedergerke, and H. Huxley and Jean Masson in England in 1954, who gave this theory as sliding filament theory. It is well supported by experimental evidence. As per the experimental evidence which supports this sliding filament theory, one of the important factors is that there is electrical and biochemical events in muscle contraction. There is electrical and biochemical events. These events take place by which the contraction of the muscles takes place. It involves complex electrical and biochemical events. These have been worked out by Albert Szent, Georgi, and Hungarian biochemist of USA, and it can be outlined as there are various things to be read first. First is the resting potential, and the second is the action potential, and the third is what we call as the muscle contraction and muscle relaxation. Third is the contraction and muscle relaxation. Resting potential and acting potential, action potential. In the resting potential, in a resting muscle fiber, the outside of the sarcolemma is positively charged. Whenever we see this sarcolemma, outside is positively charged with respect to the inside. This potential difference across the membrane is called a resting potential. Internal side is negative charge and outside is positive charge. This is sarcolemma or sarcoplasmic or the plasma membrane which surrounds the muscle fiber. It amounts to about 60 millivolts. This is 60 millivolts of potential difference. This potential difference is called as resting potential. A membrane with such a resting potential is said to be polarized. This membrane is polarized because positive charges lie outside and negative charges lie inside. The resting potential of sarcolemma is maintained by following mechanism. How this resting potential is maintained? Outside positive, inside negative. The first thing, sodium ions predominate the outside. Here the sodium ions are present outside, by which it becomes positive outside. And potassium ions predominate on the inner side. These are potassium ions which predominate on inner side. Due to different concentration on the two sides of the sarcolemma, potassium ions leave and sodium ions enter the muscle fiber. Due to differential potential, potassium ions leave. Actually, there are more sodium ions than potassium ions inside. When there is more positive charge outside, less positive charge inside, the inner side becomes negative. As a result, different concentration on two sides of the sarcolemma, potassium leaves outside and sodium ions enter the muscle fiber. Sarcolemma is more permeable to potassium ions than to sodium ions. Hence, potassium ions leave the muscle fiber faster than the sodium ions that enter into it, and this builds up the positive charge outside. By this way, the positive charge is developed outside. Sodium ions are pumped out as quickly as they enter, and potassium ions are sent in as soon as they leave, both by active transport. The process of moving ions against the concentration is called sodium pump, or better, sodium potassium exchange pump. Sodium potassium exchange pump, by which sodium ions move inside and potassium ions move outside, but the positive charges lies outside and negative charge lies inside because there are more positive ions outside than the inner side. One more thing is there, that is called as action potential. According to this, the resting potential follows the action potential. Whenever there is a stimulus, that stimulates the stimulus which leads to muscle contraction. When a muscle fiber is stimulated artificially or by a nerve impulse, the sarcolemma suddenly becomes more permeable to sodium ions. This is sarcolemma. This sarcolemma becomes more permeable to sodium ions. These sodium ions move through the sarcolemma into the interior of the muscle fiber. The sarcolemma suddenly becomes more permeable to sodium ions than to potassium ions. The potassium ions move outside, but their movement is less. Sodium ions move inside, but their movement is very great. As a result, sodium ions rush in along the concentration gradient and electrical gradient. This makes inside of sarcolemma positive with the concentration gradient and the electrical gradient. This makes inside of sarcolemma positive with respect to the outside. As a result, the charge is reversed. Firstly, in the resting potential, due to the excess sodium positive ions, due to the less potassium ions inner side becomes negative, outside positive. But when the sodium ions rush more than the potassium ions move outside, the positive charge is reversed, inner becomes positive and outer becomes negative. The sarcolemma is now to be depolarized. This stage is called as depolarization. and we can see the sarcolemma is depolarized, and this change is known as the action potential. This is called the action potential whenever there is a stimulus. That stimulus is artificially or by inner impulse. The action potential is a signal for contraction. This action potential leads to the response. The response is the contraction of the muscle. When depolarization occurs, the muscle fiber contracts. As a result, when the depolarization takes place, the muscle fiber contracts. This depolarization is the opposite of the resting potential. This is all about the muscle contraction, physiology of the muscle contraction.